Hello, Mr. Allman here, and this is your Allman Cast on semiconservative replication. The purpose of this short video is to be able to describe to you the work done by two scientists, Mendelssohn and Stahl, and how it is that they proved that the way in which DNA is copied. Now, looking at DNA structure, it's easy to see how DNA is copied. If you go back to Watson and Crick's original paper in which they described the structure of DNA, they hypothesized at that point that the complementary base pairing that exists between bases, if those two strands of DNA are separated, it will be very easy to see how an additional copy can be made by simply filling in those complementary base pairs. Now, this is called semiconservative replication. However, this wasn't proven at the time. So, there were three possible hypotheses for the way in which DNA could be replicated. The first is the semiconservative method represented here on the top. You can see that the dark red strands, those are the original template strand of DNA. In semiconservative replication, the original template strands are separated, and each of those template strands is used to produce a second new strand that is complementary to the template. The resulting is that you have in each copy of DNA that results is 50% original and 50% new. The conservative method is that original copy of DNA is going to be replicated by making a carbon copy of it. So you have an original strand, which is all that original set of DNA, and a 100% new strand of DNA. The final is the dispersive method, and in this case we have a mix of some original and some new DNA in each of those copies of DNA that results. Now, the conservative and dispersive methods, those are not found in any biologically significant amount in any type of cell. All cells are really using the semiconservative method, and the people that were able to confirm that were primarily Mendelssohn and Stahl, who in the late 1950s performed a famous experiment that supported this model of replication. Now, how did this work? If you follow the outline at the top, first they took bacteria, and they cultured bacteria in a medium that had a heavy isotope of nitrogen, nitrogen 15. Now, as the bacteria were growing and dividing, they would take in those heavy isotopes, incorporate those into their DNA. Then, those bacteria that had that heavy isotope would be put into a medium with the lighter isotope. Then, they would allow the bacteria to grow and replicate, take samples of those bacteria, put them in a centrifuge that would spin them in a very dense solution. And those that were more dense would be found further down in that centrifuge tube. Those that were less dense would rise to the top. So if we look at the results here of their experiment, in the first generation, we had 100% more dense, as they had a lot of heavy nitrogen in it. But as those bacteria with the heavy nitrogen replicated, new copies of DNA would have to be made, and because they were in a medium with that lighter isotope, they would incorporate that lighter nitrogen into their new copies of DNA. After the first generation, they saw that 100% of the bacteria had an intermediate density. They weren't as heavy as the original ones, but they weren't as light as those that were using only N14. So that shows, first of all, that it's impossible that they had a conservative replication. Otherwise, you would have seen two bands, one with the original set of DNA and one with the lighter set. But they're obviously combining them together. Now, the successive generations show that the dispersive model was incorrect, because in generations 2, 3, and 4, the amount that had that original set of DNA decreases as the amount that has those lighter isotopes increases. And we can see that from this chart. You can see what will happen after the second replication. The conservative model, after generation number 1, division number 1, is ruled out. The dispersive method, what we would expect to see is that all of the DNA decreased at a roughly equivalent amount. So you would not see separate bands. In the semiconservative model, we would have two bacteria, each with that original heavy strand of DNA, and as you can see, as those divide up again by the semiconservative method, we see that there is all light isotopes and only a smaller amount that have those original heavy isotopes. So going back to the original results, that decreasing amount, that decreasing percentage of those with N15 and the increasing amount of those with N14 shows that semi-conservative was really the way to go. So this is really an application of chemistry and some chemical separation tools as a way to show that DNA actually copies through this method. Now the remainder of this video I want to go through the process of replication and how it is that replication occurs.